Hey guys, this is our Chick Genes project. We are going to be going over DNA, Punnett squares, dominant and recessive traits, our chicks, and some other things.
Here we were dissecting the egg, and we found out that none of the eggs had hatched. This is very unfortunate, and it most likely happened because of the drought. There are many other reasons, but this is the most likely reason.
As you can see in this picture, that we're doing handling of our egg, and it shows that it is fertile. So to explain what candling is that we hold the egg up to a bright light to check if it's fertile. So if there's a large area that you cannot see through in the egg, then it is most likely fertile, which you can see right here. If there are spots on the egg or a large area you can see through, then it's not fertile.
Cells grow because of mitosis. Mitosis is when cells divide. This causes more cells, which causes growth and increase in mass. Cell differentiation is when a less specialized cell becomes a more specialized cell. Genetics happen from many different things, such as mutations, cell division, dominant and recessive traits. The traits for organisms are stored within chromosomes.
This is the gene expression, which is a conversion of the information encoded in a gene first into a messenger RNA and then to a protein. Alright, now we're going to talk about the changes in DNA. Everyone acquires some changes in their DNA during the course of their lives. These changes occur in a number of ways. Sometimes there are simple copying errors that are introduced when DNA replicates itself. Other changes are introduced as a result of DNA damage through environmental agents, including sunlight, cigarette smoke, and radiation.
Our cells acquire a lot of damage throughout our lives, but they have built-in mechanisms that catch and repair most of it. So while we get older, we develop more and more cell damage because it just can't do all of it. So yeah, when you get older, you have more cell damage.
Meiosis produces the gametes, sperm in the male and egg in the female. It is important and distinct from mitosis in that the cells it produces are haploid. They have half the usual number of chromosomes. Gametes must be haploid to ensure that they combine to form a zygote, fertilized egg. It has the correct number of chromosomes. Transcription. To create mRNA, the DNA is unwound and then attaching complementary growing mRNA molecules. Uracil is substituted for thymine and as the complementary for adenine. Translation. After the mRNA leaves the nucleus, the complementary strand is added again.
DNA fingerprinting is a shortcut to mapping genomes and DNA sequences. Using this method, genomes can be compared to other genomes by looking at the bars on the graph and seeing if they match up. This is also used to see genetic modifications by comparing DNA sequences that are a part of the same genome and observing to see if any modifications occurred.
This is a Punnett square of a Rhode Island red and a barred rooster. The Rhode Island red is the dominant parent, and the barred rooster is the recessive parent. There would be 0 homozygous dominant, 1 heterozygous dominant, and 3 homozygous recessive offspring in this Punnett square.
The phenotype is 3 barred and 1 Rhode Island red.